So in our last YouTube video, we looked at Mendel's genetics, and now we're going to look at non-Mendelian inheritance, which includes three main things: epistasis, co-dominant, and incomplete dominance. These are the three rules that Mendel's ideas didn't follow. So first of all, we'll be looking at incomplete dominance. So this is where there is no dominant gene or dominant alleles that does not, or the dominant allele does not mask the recessive allele. The phenotype will have come from neither parent, so it won't look the same as either parent, but it will show a mixture of both parents. So for an example is this, that you'll see in pretty much every textbook, is red and white flowers. So these show incomplete dominance. They show the primary generation, the parental generation that are mixed together. So with capital R and little r. Now capital R is the dominant, is supposedly the dominant allele, and white R is less dominant, but neither show full dominance, so they are mixed together and form a pink flower, which looks like neither phenotype of the parents. Now we look at co-dominance. So this is where both alleles are expressed at once. So both show up in different quantities. So for example, the phenotypes are distinctly mixed. You can see this cat, half of it, well, majority of it is black, and then the other section is brown. And they don't mix, the colors don't mix. So they're as dominant as each other in a sense that it will either be one color or the other in a specific location. So they won't mix. They'll be distinctively be able to be separated. Now we're looking at epistasis, the third and final non-Mendelian theory. So this is where one expression in a gene changes the expression of a different gene. So we're going to look at the example of Labradors. So for the example in Labradors, there is a gene to say if there will be a pigment shown, so the color of the dog. If not, if there's no pigment, then it'll be yellow. So then the next gene will determine if it'll be black or brown. So basically what this is saying is that the gene will either, if the gene is present to show pigment, the pigment can then be an option of black or brown. I'll show there in the next slide. So here we have a dihybrid cross with more than one allele. So we're looking at E, which represents will pigment be shown, capital E being yes, being the dominant, and then we also have B, capital B being black, which is dominant, and little b being brown. So if you have two small b's, then it will be chocolate, and then if you have a homozygous or heterozygous with a capital B, then it will be black. However, if you have two little, it's the same with E's. If there's a one or two big E's, it will show the pigment. However, if you have two little E's, then no pigment will be shown. So even if it was two little E's, which no pigment will be shown, and then the allele for black, it will still be yellow. Similar with if it's no pigment is shown and the allele will show brown, then no pigment will be shown. Thank you very much.